So hello, so hello everyone. Today we are going to study Unit 9, that is Biotechnology. And it consists of two chapters: Biotechnology Principles and Processes, and Biotechnology and its application. Ever since the days of René Descartes, the French philosopher, mathematician, and biologist of 17th century, all human knowledge, especially natural sciences, were directed to develop technologies which add to the creature comfort of human lives, as also value to human life. The whole approach to understanding natural phenomena became anthropocentric. Physics and chemistry gave rise to engineering, technologies, and industries which all worked for human comfort and welfare. The major utility of the biological world is as a source of food. Biotechnology, the 19th century pursuit of modern biology changed our daily life as it product brought a qualitative improvement in health and food production. The basic principle underlying biotechnological processes are some application and highlighted and discussed in this unit. Herbert Boyer was born in 1936 and brought up in a corner of western Pennsylvania where railroads and mines were the destiny of most young men. He completed graduate work at the University of Pittsburgh in 1963, followed by 3 years of postgraduate studies at Yale. In 1966, Boyer took over assistant professorship at the University of California at San Francisco. By 1969, he performed studies on a couple of restriction enzymes of E. coli bacteria with especially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strand in a particular fashion, which left what has become known as sticky ends on the strands. These clipped ends made pasting together pieces of DNA a precise exercise. This discovery in turn led to a rich and rewarding conversation in Hawaii with a Stanford scientist named Stanley Cohen. Cohen had been studying small ringlets of DNA called plasmids and which float about freely in the cytoplasm of certain bacterial cells and replicate independently from the coding strand of DNA. Cohen had developed a method of removing these plasmids from the cell and then reinserting them in other cell, combining this process with the DNA splicing enabled Boyer and Cohen to recombine segment of DNA in desired configuration and indulge the DNA in bacterial cells, which could then act as manufacturing plant for specific protein. This breakthrough was the basis of which the discipline of biotechnology was founded. Chapter 9: Biotechnology Principles and Processes. Biotechnology deals with techniques of using live organism or enzymes from organism to produce products and processes through humans. In this sense, baking curd, bread, or wine, which are all microbes mediated process, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organism to achieve the same on a larger scale. Further, many other processes or techniques are also included under biotechnology. For example, in vitro fertilizers leading to a test tube baby synthesis gene and using it, developing a DNA vaccine or correction of defective genes are all part of the biotechnology. The European Federation of Biotechnology has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFP is as follows: the integration of natural sciences and organism cells, part thereof, and molecular analogs of for products and services. Principles of biotechnology. Among many, the two core techniques that enabled birth of modern biotechnology are genetic engineering techniques to alter the chemistry of genetic material DNA and RNA to introduce these into host organism and thus change the phenotype of the host organism. The second is bioprocess engineering, maintenance of sterile microbial contamination or free ambience in chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological product like antibiotics, vaccines, enzyme, etc. Let us now understand the conceptual development of the principle of genetic engineering. You probably appreciated the advantages of sexual reproduction over asexual reproduction. The former provide opportunities for variation and formulation of unique combination of genetic setups, some of which may be beneficial to other organisms as well as the population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. The traditional hybridization procedures used in plants and animal breeding very often led to inclusion and multiplication of undesirable genes along with the desired gene. The techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning, and gene transfer, overcome this limitation and allows us to isolate and introduce only one or a set of desirable genes. without introducing undesirable genes into the target organism. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of a chromosome which has the ability to replicate in chromosome. There is a specific DNA sequence called the origin of replication which is responsible for initiating replication. Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be a part of chromosome which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA was from the possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA, the Salmonella typhimurium. Stanley Cohen and Albert were accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from the plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of the so-called molecular scissors restriction enzyme. The cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which acts which acts on cut DNA molecules and join their ends. This makes a new combination of circular autonomously replicating DNA, creating in vitro, and is known as recombinant DNA. When this DNA is transferred into Escherichia coli bacterium closely related to Salmonella, it could replicate usually using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. You can hence infer that there are three basic steps in genetically modifying an organism: identification of DNA with desirable genes, introducing of the identified DNA into the host, maintenance of the introduced DNA in the host, and transfer of DNA to its progeny. Tools of recombinant DNA technology. Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools. It means restriction enzymes, polymerase enzyme, ligase, vectors, and the host organism. Let us try to understand some of these in detail. Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease in the second whose functioning dependent on a specific DNA dependent on a specific DNA nucleotide sequence was isolated and characterized. 5 years later, it was found that hens can always cut DNA molecules at a particular point by recognizing a specific sequence of six base pair. This specific base pair sequence is known as the recognition sequence for hen second. Beside hen second, today we know more than 900 restriction enzymes that have been isolated from over to 30 strains of bacteria, each of which recognize different recognition sequences. The convention of for naming these enzymes is the first letter of the name comes from the genus and the second two letters come from the species of the prokaryotic cell from which they were isolated. For example, E. coli comes from Escherichia coli RY13. In E. coli, the letter R is derived from the name strain. Roman number are following the names indicating in the order in which the enzymes were isolated from that strain of the bacteria. Restriction enzymes belong to a larger class of enzymes called nucleases. These are of two kinds, in exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at specific positions within the DNA. Each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Do you know what palindromes are? These are groups of letters that form the same words when read both forward and backward. For example, Malayalam. As again, the word palindrome, where the same word is read in both directions. The palindrome in DNA is a sequence of base pairs that read same on the two strand, where orientation of reading is kept the same. For example, the following sequences read on the two strand 5 dash to 3 dash, and this is also true if we read in 3 dash to 5 dash direction. Restriction enzymes cut the strand of DNA a little away from the center of palindrome sites but between the same two bases on the opposite strand. This leaves single stranded portion at the end. There are overhanging stretches called sticky ends on the strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitates the reaction of enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and then these can be joined together end to end using DNA ligase. You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease result in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move toward the anode under the electric field through a medium or matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from seaweeds. The DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller fragment size, the farther it moves. Look at the figure 9.3 and guess at which end of the gel is sample was loaded. This is figure 9.3 and answer the question given here. The separated DNA fragments can be visualized only after staining the DNA with the compound known as ethidium bromide followed by exposure to UV radiation. You cannot see pure DNA fragment in the visible light, and without staining you can see bright orange colored bands of DNA in the ethidium bromide stained gel exposed to UV light. The separated band of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. The DNA fragments are purified in this way are used in the constructing recombinant DNA by joining them with cloning vectors. You know that plasmids and bacteriophages have the ability to replicate within bacterial cells independent of the control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy number of their genome within the bacterial cells. Some plasmids may have only one or two copies per cell, whereas others may have 15 to 100 copies per cell. Then their numbers can go even higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of a plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. Following are the features that are required to facilitate cloning into a vector. Origin of replication. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cell. These sequences are always also responsible for controlling the copy number of the linked DNA. So if one want to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. Selectable marker. In addition to the vector requires a selectable marker which help in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. You will study the process in subsequent section. Normally, the genes encoding resistant to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Cloning sites. In order to link the alien DNA, the vector needs to have very few preferably single recognition sites for the commonly used restricted enzyme. Presence of more than one recognition sites within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at the BAMH first site of tetracycline resistant gene in the vector PBR322. The recombinant plasmid will lose tetracycline resistance due to the insertion of foreign DNA but can still be selected out from non-recombinant ones by plating the transformation on ampicillin containing medium. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on the containing tetracycline, but non recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistant gene gets inactivated due to insertion of the alien DNA and helps in selection of recombinants. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating onto plates having different antibiotic. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of a chromogenic substrate gives blue color colonies if the plasmids in the bacteria does not have an insert. Presence of insert result into insertional inactivation of the beta galactosidase gene, and the colonies do not produce any color. These are identified as recombinant colonies. Vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the relation of transferring genes into plants and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cells and for them to do what the bacteria or viruses wanted. For example, Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce a chemical required by the pathogen. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors while delivering genes of interest to humans. The tumor-inducing TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plant but but is still able to use the mechanism to deliver genes of our interest into a variety of plants. Similarly, retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into the bacterial, plant, or animal host where it multiplies. Competent host for transformation with recombinant DNA. Since a DNA is a hydrophilic molecule, it cannot pass through cell membrane. Why? In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up DNA. This is done by treating them with a specific concentration of divalent cation, such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cell by incubating the cells with the recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat soak and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cell. In a method as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. Another method suitable for plant cells are bombarded with high velocity microparticle of gold or tungsten coated with DNA in a method known as biolistics or gene gun. And the last method uses disarmed pathogen vectors, which, when allowed to infect the cell, transfer the recombinant DNA into the host. Now that we have learned about the tools for construction recombinant DNA, let us discuss the processes facilitating recombinant DNA technology. Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in a specific sequence, such as isolation of DNA, fragmentation of DNA by restriction endonuclease, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cell in medium at a large scale, and extraction of desired product. Let us examine of each of these steps in some detail. Isolation of genetic material, then cutting out DNA at a specific location, amplification of gene of interest using PCR, and then insertion of recombinant DNA into the host cell, obtaining a foreign gene product, downstream processing, and now chapter will be finished. So let us study each of these. Recall that nucleic acid is the genetic material for all organisms without exception. In majority of organisms, this is deoxyribonucleic acid or DNA. In order to cut the DNA with a restriction enzyme, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, proteins, polysaccharides, and also lipids. This can be achieved by treating the bacterial cell or plant or animal in tissue with enzymes such as lysozyme, bacteria, cellulase, plant cell, chitinase, fungus. You know that genes are located on long molecule of DNA intertwined with proteins such as histones. The RNA can be removed by treatment with a ribonuclease, whereas proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatments, and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as collection of fine threads in the suspension. Cutting of DNA at specific locations. Restriction enzyme digestion are performed by incubating purified DNA molecules with the restriction enzyme at the optimal condition for that specific enzyme. Agarose electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves toward the positive electrode. The process is repeated with the vector DNA also. The joining of DNA involves several processes after having cut the source DNA as well as the vector DNA with the specific restriction enzymes. the cutout gene of interest from the source of DNA and the cut vector with the space are mixed and ligase is added. This results in the preparation of recombinant DNA, amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene of interest is synthesized in vitro using two sets of primes, small chemically synthesized oligonucleotides that are complementary to the regions of DNA and the enzyme DNA polymerase. The enzyme extend the primers using the nucleotides provided in the reaction and our genomic DNA as a template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. It means 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from the bacteria Thermus aquaticus, which remain active during the high temperature induced denaturation of the double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Insertion of recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA into recipient cells. Recipient cells, after making them competent to receive, take up DNA present in its surroundings. So, if a recombinant DNA bearing gene for the resistant to an antibiotic, for example, ampicillin, is transferred into E. coli cells, the host cell become transferred into ampicillin resistant cells. If we spread the transformed cells on agar plates containing ampicillin, only transformants will grow. Untransformants recipient cells untransformed recipient cells will die since due to ampicillin resistant gene one is able to select a transformed cell in the presence of ampicillin. The ampicillin resistant gene in this case is called a selectable marker. Obtaining the foreign gene product, when you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant or animal, so the alien DNA gets multiplied in almost all recombinant technologies. The ultimate aim is to produce a desirable protein, hence there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate condition. The expression of foreign genes in host cells involve understanding many technical details. After having cloned the DNA of interest and having optimized the condition to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in heterologous host, it is called recombinant protein. The cell harboring cloned gene of interest may be grown on a small scale in the laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. The cells can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their uh, physiologically most active log or exponential phase. This type of culturing method produces a larger biomass, leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors, where large volumes 100 to 1000 liter of culture can be processed, was required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc., using microbial plants, animals, or human cells. A bioreactor provides the optimal condition for achieving the desired product by providing optimum growth condition, temperature, pH, substrate, salt, vitamin, or oxygen. The most commonly used bioreactor are of stirring type, which are shown in figure 9.7. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor content. The stirrer facilitates even a mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivery system, and a foam control system, a temperature control system, pH control system, and sampling ports to that small volumes of the culture can be withdrawn periodically. Downstream processing. After completion of biosynthetic stage, the product has to be subjected for a series of processes before it is ready for marketing as a finished product. The processes include preparation and purification, which are collectively referred to as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo thorough clinical trials, as in case of drugs. Strict quality control testing of each product is also required. The downstream processing and quality control testing vary from product to product. Summary: Biotechnology deals with the large scale production and marketing of products and processes using live organisms, cells, or enzymes. Modern biotechnology using genetically modified organisms was made possible only when man learned to alter chemistry of DNA and construct by recombinant DNA. This key process is called recombinant DNA technology or genetic engineering. This process involves the use of restriction endonuclease, DNA ligase, appropriate plasmid or viral vectors to isolate and ferry the foreign DNA into host organism. Expression of the foreign gene, purification of the DNA product, it means the functional protein, and finally making a suitable formulation for marketing. Large scale production involve use of bioreactor. So our chapter is finished.